Hi, I am Dr. Arsalan and today we are going to solve the MCQs on cell. First question is, which of the following organelle lacks DNA? Given options are nucleus, mitochondria, chloroplast and centrosome. It should be kept in mind that DNA is present in the chromosomes and gene in the nucleus. As well as the mitochondria and chloroplast, these are the organelles which are thought to be the prokaryotic cell at the earlier stage. These were entered and innervated in the cells through the process of endosymbiosis. So mitochondria and chloroplast have their own DNA and ribosomes, and these are self-replicating bodies. So DNA is present in nucleus, mitochondria, chloroplast, but centrosome lacks the DNA. So option D is correct, that centrosome are the organelles which lacks DNA. Second question is, cell theory doesn't imply on. Given options are virus, bacteria, fungi and plants. Cell theory was presented by Schleiden and Schwann in 1838 and 1839, in which they stated that all living things are made up of cells. So the organisms which lack cellular machinery are those organs which are acellular in nature, are not under the scope of this cell theory. So the viruses are acellular structures, acellular organisms having incomplete cellular machinery. So cell theory does not implement on the virus. Option A is correct, that the virus are the organisms on which the cell theory does not implement. Next question is, glycolipids are located on dash of plasma membrane. The given options are inner leaflet, outer leaflet, both or none. As we know that in the cell membrane there is phospholipid bilayer in which the proteins are penetrated, called intrinsic proteins. These intrinsic proteins extend from one end of the phospholipid to the other. For example, this is phospholipid bilayer and there are proteins which extending from one end to the other end. These are called intrinsic proteins, and some proteins are present on the surface of this phospholipid bilayer. These are called extrinsic proteins. And then the carbohydrates are attached on both the lipid bilayer as well as the proteins. So the carbohydrates which are attached to the protein are called glycoproteins, and the carbohydrates which are attached to the lipid bilayer are called glycolipids. So these glycolipids, these are attached to the outer surface of the plasma membrane or the phospholipid bilayer. So the option A is inner leaflet and option B is outer leaflet. So this option is correct, that these glycolipids or the carbohydrates attached to lipids are present on the outer surface of the phospholipid bilayer or plasma membrane. Next question is, dash and hydrolase enzymes are present in lysosomes. Given options are catalase, oxidase, lyase, acid phosphatase. Lysosomes are the cytoplasmic organelles which are sac-like structures present in the cytoplasm containing hydrolytic enzymes. These are involved in the phagocytosis and cell killing process. So have the hydrolytic enzymes. There are two types of hydrolytic enzymes present in the lysosomes. These are hydrolase and acid phosphatase. So hydrolase is given in the question and the option D is acid phosphatase. So these two enzymes are present in the lysosomes. Acid phosphatase and hydrolase are the enzymes which are present in the lysosomes, which break down the foreign organisms which enter the cells by the process of phagocytosis. The question is, cell without cell wall is called. So given options are tonoplast, leucoplast, apoplast and protoplast. Tonoplast is the membrane of vacuole. So this is not correct option. Leucoplast is the colorless plastid. Apoplast is a dead pathway of transport of water inside the roots. So all these options are incorrect. The final option, protoplast, is the cell lacking the cell wall. Or the cell without cell wall, it is called protoplast. So the option D is correct. Next question is, the fluidity of plasma membrane increases with increase in dash. Given options are unsaturated fatty acids, saturated fatty acids, proteins and glycolipids. As we know that the cell membrane or plasma membrane, it is also called as sea of lipids. So the fluidity of the cell membrane is due to lipids, the phospholipid bilayer. Two kinds of lipids, unsaturated fatty acids and saturated fatty acids. Unsaturated fatty acids are those which are liquid at room temperature. For example, the alls, these have double and triple bonding between the carbon atoms, while saturated fatty acids have single carbon to carbon bonds and these are solids at room temperature. For example, the fats. So the fluidity is increased with the oils because oils are liquids and these are unsaturated fatty acids. So the option A is correct, that the unsaturated fatty acids increase the fluidity of the plasma membrane. Next question is that the apoptosis is done by. Given options are ribosome, centrosome, lysosome and peroxisome. Apoptosis means the programmed or normal cell death in which the abnormal, worn out, broken and damaged body cells are removed from the body or killed by the process of phagocytosis by the lysosomes. This process is called autophagy. Apoptosis is done by the lysosomes. This process is called autophagy, in which the lysosomes eat out its own worn out, damaged and unwanted cells of the body. The programmed cell death, the apoptosis. While rest of the ribosomes, these are involved in the protein synthesis. Centrosome are involved in the cell division. The peroxisomes are involved in the neutralization of hydrogen peroxide into water and oxygen. Have no concern with the apoptosis or cell death, or the lysosomes, being suicidal bags, attack its own body cells. This process is called apoptosis. Next question is, in DASH cells, gap junctions are absent. Given options are sperms, cardiac cells, hepatocytes, or muscle fibers. Gap junctions are basically the spaces between the cells in which the neurotransmitters carry the impulse from one cell or one neuron to the other cell. So junctions or spaces are required for the transmission of neuron impulses from the one cell to the other cells through neurotransmitters. So the option sperms is correct because sperms don't have gap junctions. It should be kept in mind that any of the motile cells lack gap junctions. There are no gap junctions between the moving cells. Gap junctions are only present in the stagnant or stationary cells present in the body in a fixed position, so that the nerve impulse may be transmitted from one cell to other cell. Next question is, the sarcoplasm is the cytoplasm of DASH. Given options are mitochondria, plant cell, muscle fiber, or all of these. The sarco word is basically used for muscle fibers because the cytoplasm of muscle fiber is called sarcoplasm. The endoplasmic reticulum of the muscle fiber are called sarcoplasmic reticulum. Option C is correct, that the muscle fibers have the cytoplasm, which is called as sarcoplasm. Next question is, DASH produce the lysosomes. Lysosomes are these cytoplasmic organelles, as we have discussed, that these are the suicidal bags. These have hydrolytic enzymes for killing off any of the foreign object which enter the cells. So these are synthesized basically by the given options are endoplasmic reticulum, Golgi bodies, nucleus, or cell membrane. It should be kept in mind that the endoplasmic reticulum synthesizes all the proteins and the enzymes which are stored and packaged and modified by the Golgi bodies in the form of vesicles. Then at the time of need, these Golgi bodies let their vesicles to be budded off from their dictyosome, and these budded off the vesicles are called lysosomes. So these are basically synthesized by the Golgi bodies. Option B is correct. Cell wall of bacteria is made up of DASH. Given options are murine, peptidoglycan, N-acetyl glucosamine, are all of these. As we know that the cell wall is present in three kinds of organisms, three kingdom of organisms, the fungi, plants, and kingdom prokaryote or bacteria. In plants, the cell wall is made up of cellulose and hemicellulose. In the fungi, cell wall is made up of chitin. These are all carbohydrates. In case of bacteria, the cell wall is made up of murine, which is also called as peptidoglycan, which is composed of acetyl glucosamine or N-acetyl glucosamine. So these all options pertain to the cell wall of the bacteria. So option D is correct, that all of these are correct options. The cell wall of bacteria is made up of murine, which is also called as peptidoglycan, which is made up of or composed of unit of acetyl glucosamine. Next question is that rRNA is synthesized in. Given options are endoplasmic reticulum, Golgi bodies, nucleus, and nucleolus. The rRNA refers to the ribosomal RNA. There are three types of the RNA: the transfer RNA, the ribosomal RNA, and the messenger RNA. Here is asking about the ribosomal RNA, which constitutes or make up the ribosomes along with its protein components. So these are synthesized in endoplasmic reticulum. So option is incorrect. Neither in the Golgi bodies. These are synthesized inside the nucleus in the nucleolus. So both options are correct, but the most specific one is the nucleolus. That the nucleolus are darkly stained spherical structures present in the nucleus without having any membrane, and these are required for the synthesis of ribosomal RNA. These have the precursors in its external or peripheral granular region. For nucleolus have two regions. The region is called peripheral granular region, in which there are precursors of ribosomes, and the inner region is called central fibrillar region. In case of peripheral granular region, there are precursors of ribosomal RNA which synthesize the ribosomal RNA or rRNA. So the option D is correct. Nucleolus are responsible for the synthesis of ribosomal RNA. The question is, DASH is also called mitoplast. So this is a new word, mitoplast. The given options are mitochondria without the membrane, mitosis, plastids containing mitochondria, or none of these. So the option A is correct, that the mitochondria without the membrane are called mitoplast. Like the cell without cell wall is called protoplast. Similarly, the mitochondria without the wall or membrane is called mitoplast. Other process is mitosis. This is cell division process. Plastids containing mitochondria. So the plastids are separate organelles. Then the mitochondria, there are no mitochondria inside the plastid. So this option is also incorrect. And the last is none of these. So the correct option is that the mitochondria without or lacking the membrane are called mitoplast. So this next question is, sodium potassium pump is the example of DASH. Given options are active transport, passive transport, diffusion, or osmosis. Sodium potassium pump is basically involved for the transmission of nerve impulses across the cells. We learned this topic in nurse coordination. So this is the example of active transport. Option A is correct. Next question is, macrophage is a dash cell. Given options are anucleate, mononucleate, binucleate or multinucleate. Cells having no nucleus are called anucleate. Cells having single nucleus is called mononucleate. With two nuclei, the cell is called binucleate. And cell with multiple or many nuclei are called multinucleate cells. So macrophage is large cell. It is made from the monocytes. When many monocytes combine together, they form a large macrophage cell for the immunity purpose. And this is a multinucleated cell. The option D is correct. The last question is, RBCs lack. RBCs are the red blood cells which are deficient or which are lacking the cell membrane, cytoplasm, nucleus, or all of these. So this is a fact based question that RBCs always lack the nucleus. They are anucleate cells. So the option C is correct. The RBCs lack nucleus. Hope this video will help you a lot in preparing for your medical colleges admission tests, and don't forget to share your marks, that how many MCQs you are going to solve in this lecture in the comments box, and how many MCQs you have learned in this lecture.